Good morning, good afternoon, good evening, everybody. It's my pleasure to welcome you to another talk in the series of Global IMMUNOTALKs 2023. I'm Laura McKay. I'm based at the Doherty Institute in Melbourne, and it's a privilege to introduce this week's speaker on behalf of our 2023 Global IMMUNOTALKs organizing team. Now, our speaker today, Jose Villadangos, is coincidentally in the same building as me right now. We're pre-recording from Melbourne in Australia. And this is where Jose holds two appointments, both at the Department of Microbiology at the Peter Doherty Institute and also at the Department of Biochemistry and Pharmacology at the Bio21 Institute, both at University of Melbourne. Now, Jose, which will become evident when Jose starts to deliver his lecture, is originally from Spain. He obtained his PhD originally from the University of Madrid, and then subsequently Jose went halfway across the world to MIT and Harvard Medical School, where he was trained further by Edie Pla, and then Ken Shortman at the Walter and Eliza Hall Institute in Melbourne. And it's here where Jose started his lab. That was back in 2001 now. And this is really where Jose is best known for his seminal contributions to the fields of antigen presentation and T-cell recognition and dendritic cell development. Now, Jose has now published over 150 articles. He's editor-in-chief in 2 journals, both Molecular Immunology and Current Research in Immunology. And perhaps one of Jose's notable contributions of many was he was president of the International Congress of Immunology, the ICI meeting, that was held here in Melbourne in 2016, which was really a heroic effort. And Jose was recipient of the Derek Rowley Medal of the Australian and New Zealand Society for Immunology, where Jose is a lifetime member. Now, Jose's laboratory has a broad interest ranging from antigen presentation by classical and non-classical MHC molecules. He's interested still in dendritic cell development and function, adoptive T-cell therapy against cancer and antiviral immunity. And so perhaps Jose's talk is very aptly titled The End of Antigen Presentation and the Beginning of Adaptive Immunity. And so, Jose, if you could switch on your camera, we would love to welcome you to Global IMMUNOTALKs. We are so happy that you accepted the invitation here to speak. And as you know, one of our traditions here at Global IMMUNOTALKs is to get to know you a little better. Now, I can definitely attest that Jose is one of the most fantastic mentors around, really being a treasure to many of the trainees that goes through his laboratory. And so, Jose, we'd like a little advice from you. Many of us are immigrants in where we live now and where we do science, and you as an immigrant yourself here in Melbourne. We'd like to know, what advice would you give to new trainees that are relocating to a different country and embarking on their scientific journey?
Thank you, Laura. Well, I have been an immigrant in 2 different countries, first in the US and in Australia. And I think that the main piece of advice that I will have for any scientist who becomes an emigre is not to stop thinking about returning home, at least for a while, and be open about the opportunities that you will find in your new home country. Those opportunities will be scientific, and you will be exposed to new ways of organizing laboratories and funding new collaborators and new possibilities for extending your research. But the other, of course, is the opportunity to learn more about the culture of the new place and get to know the physical and social aspects of the country that you visit, its politics. And of course, I could get a lot of this over my years first in the United States and then in Australia. That would clearly be one of the top advice that I have. Don't think too much, or not all the time, at least, on work. There will be opportunities for you to travel and know the country. So do that, because the postdoc is the period in the career of a scientist that offers more opportunities for personal enrichment. So take them.
Thank you so much, Jose. And that's such great advice from someone that successfully lived on 3 continents of the world. And of course, you know, that's such an apt answer here on the Global IMMUNOTALKs forum, where, you know, immunology, it's such a global Korea, you can really live anywhere. So thank you so much, Jose. We're so looking forward to your talk. So if you could share your slides and then take it away for us.
Going for it now, Laura. So Laura, it's all looking the way it should.
Yes, that's absolutely perfect, Jose. Thank you.
Okay. First of all, thank you, Laura, and thank you to the organizers for this opportunity to talk in global IMMUNOTALKs. My introductory slide shows the two buildings where the two sections of my laboratory are located. I do research on immunology. I also do research on biochemistry and molecular biology that uses the immune system as a model. And today I would like to showcase two different areas of study in my laboratory that are good representative of the research that we do, more biased on immunological aspects and more biased on biochemistry and cell biological aspects. So I'm taking the concept of global in two ways. First, as an opportunity to reach the globe via online posting of a video, but also with the mindset of this being open to a large audience of immunologists, both non-immunologists as well, both experts who have spent already a significant number of years in the field, and newcomers, new PhD students, and young new postdocs who may want to gain a more broad understanding of immunology. So let me start with a relatively basic description of what happens when an organism becomes infected with a pathogen. So what is going to happen is that a number of cellular players will interact with each other in secondary lymphoid organs that will lead to the formation of a response. You can study this process at a more detailed level, looking at the interaction between two cells. But the interaction of those two cells is founded on the processes that are happening inside the cells. And this square there represents what has been the area of my research for over 30 years, the intracellular processes that lead to the display of MHC peptide complexes on the surface of an antigen-presenting cell, in other words, antigen presentation. During an immune responses to either a pathogen or a vaccine, all these events are going to happen, and they will lead to the formation of first an innate immune response and then an adaptive immune response. During the initiation of adaptive immunity, only a small number of dendritic cells, which contain in lymphoid organs, present pathogen antigens to T cells. The rest only present self. Of those dendritic cells that present antigens, only some of the MHC molecules that are exposed on the surface of the antigen-presenting cell will be displaying pathogen antigens. The rest of the molecules will be presenting the self-protein. So this already shows you the challenge of initiating an adaptive immune response. These rare dendritic cells in lymphoid organs that are in charge of initiating the response that will be presenting rare MHC peptide complexes on their surface, I will need to interact with rare T cells that have the correct T cell receptor to recognize the MHC peptide complexes derived from the pathogen. All these conditions need to be met for an adaptive immune response to start. So insofar the life of this organism now depends on its ability to initiate an adaptive immune response against a pathogen, its life now depends on the efficacy of this process. This is what will trigger a familiar process over time of initiation of the response I had just described here, that will lead to the expansion of lymphocytes that will become effector cells that will be able to produce antibodies in the case of B cells or do other things in the case of T cells, which hopefully will lead to the elimination of the threat and will be followed by the contraction of the response, leaving behind only a small number of memory cells capable of responding to the next challenge. This whole process can be collapsed in a single, more simplified bell curve that looks like this. And this bell curve shows you how an immune response develops, comes back at the end of the infection to a baseline, to return to homeostasis. What I'm going to show you first in this talk is situations, what happens when this is not the actual outcome of the response and things don't go exactly like this. Indeed, it is very likely that immune responses never quite undergo this neat process of return to homeostasis as we described here. I'm going to talk about the consequences for the immune system of sepsis. And to do that, I would first like to elaborate on the definition of sepsis, because this is a word that is commonly used by immunologists, but not always used in the way that is officially understood by clinicians. In very common textbooks of immunology, I put this one as an example. It could be different, like the Abbas. Sepsis is described as a bacterial infection of the bloodstream. And one of the consequences of this can be septic shock. This is an over response of the immune system that causes organ damage. And the most common model to mimic this process in experimental animals is the Zika ligation puncture model, which is a good model for septic shock. Sepsis in the clinic, and sepsis more understood as an experimental problem, is wider than this. So there is a number of clinical definitions to say when you have sepsis. But to summarize it all, you can describe sepsis as a severe infection by any pathogen. It doesn't have to be by bacteria. It can be caused by viruses, or it can be caused by fungi. Anywhere in the body, it doesn't have to happen in the blood. It can be in your lungs. That makes the immune system go local. This is a bit out of control. And in our graph before, this can be represented by a red line that instead of stopping and as in a normal immune response, keeps going, producing an exacerbated immune response that is what we call, the now familiar for everybody who has been on this planet for the past 3 years, a cytokine storm like the one we have experienced in severe cases of COVID-19. And this causes multiple organ damage that can cause death. We have been interested in some of the ways that the immune system can contribute to this organ damage, which is not only caused is not only triggered by infections. Sepsis is a form of a wider term, which is SIRS, Systemic Inflammatory Response Syndrome. There are a number of conditions that can cause the immune system to become uncontrolled, as it happened during sepsis. And these are just associated with situations that cause strong and severe inflammation, as it can be a severe trauma and certain surgeries. In particular, we published last year a paper in collaboration with a group of Karima Senoune in Nantes that describes a mechanism that can be responsible for kidney damage during SIRS caused, in this case, by surgery. This is a mechanism of destruction of kidney cells mediated by neutrophils by a process of exacerbated trogocytosis, a term that I will use later on in this talk. During this process, cells capture small fragments from other cells. And when they do that in large amounts, they can cause the death of the cell they are trogocytosing. And this is what is termed trogoptosis. So this is an example of a mechanism that the immune system can use to cause damage to your organs in intensive care. Fortunately, modern medicine can help to overcome this damage and cause the survival of patients. However, even if a patient survives this critical period of cytokine storm and organ damage, the patient is not out of the woods, because then something interesting happens. Even though the immune system of this patient is undergoing high activity, the patient becomes susceptible to secondary or opportunistic infections, what is generally called hospital-acquired infections, of which the most common is pneumonia, so infection of the lungs. This is a big problem. So here are some figures that you can see the number of episodes of hospital-acquired infections that happen, or often associated with patients that are in intensive care. This is a very costly management to have in intensive care. This is the reason why virtually every patient who is admitted to intensive care unit is treated with antibiotics, even if the intensivists are not sure that the patient is suffering SARS caused by bacterial pneumonia. And in fact, COVID-19 patients are treated with antibiotics as soon as they get to the intensive care as a prevention against secondary infection with bacteria. You know that antibiotics are not effective against a virus. While this may be effective in contributing to the prevention of the secondary infection, it is a contributor to the emergence of antibiotic resistance. So it would be good to be able to find alternatives to the problem. And in recent years, and over time, I think that one of the pioneers of this idea was Hotchkiss in St. Louis. There has been an emergence of the concept that one of the reasons why the patients are susceptible to secondary infections is not just that they are weakened or that their mucosas are damaged by the systemic inflammation, but that they are actually immunosuppressed and incapable of responding to infections that a non-patient, a healthy person, would not have to worry about. A number of years ago, we started to study this phenomenon, trying to use different models of infection that cause systemic inflammation to characterize to what extent there is immunosuppression in these conditions and to what extent that can be attributed to defects in antigen presentation and the activity of antigen-presenting cells, in particular dendritic cells. One of my favorite models for this is malaria infection, which we started to use in collaboration with Brendan Crabb and Bill Heath while I was still at the Walter and Eliza Hall Institute. And I continue to use these days with Bill Heath at the Doherty Institute. If you infect a mouse with the Plasmodium parasite, in this case Plasmodium bergei, you can measure the presence of the parasite in blood. That's represented here. It increases over time. At a certain point, the amount of parasite is so that it triggers systemic inflammation in the mouse. And this can be measured by measuring, as Mitra Shajaripana did in this study here that we have in preparation. You can measure the amount of different cytokines shown at the top over time following infection. And what you can see is approximately at day 4, you start to see the levels of cytokine, several cytokines in serum increasing. First is some, then are others, and so on. Subsequently, this clears, as I will show you later on. But what this shows is that this is a situation where the infection triggers systemic activation of the immune system in the mice. And one of the questions that we ask is, what is the immunocompetence of these mice in this situation? And we can ask that by injecting a vaccine into the mouse, a model vaccine that in this case consists of the protein ovalbumin, and measuring the response to this protein. And what we observe is that as the parasitemia increase in this mouse, as the malaria infection progressed, the mouse became less and less capable of responding to the infection, to the point that by day 4, the immune system of these mice could not respond to the vaccination. In these mice, there were no cells capable of telling the T cells that they should respond against a new antigen injected into that animal. This can be mimicked by other ways. A much simpler way of doing it is just by injecting a TLR ligand, as shown here, with CpG or LPS. The difference with malaria is just in a few hours, just 4 hours after injecting any of these TLR ligands, you get a very clear systemic inflammation in the mouse that can be measured as cytokines in serum. And again, you can ask what is how good is the response after giving that challenge, and you can try it with different challenges. Here are examples where we challenge the mice with 2 different viruses in 2 different strains of mice, and it didn't matter what was the trigger of the systemic inflammation. These mice treated this way could not tell their immune system that they had been infected with a virus subsequently. They couldn't mount an adaptive immune response against the virus. And the reason was because their dendritic cells were incapable of presenting the antigens encountered following the systemic inflammation. We knew that the dendritic cells had been systemically activated because just by measuring the levels of MHC class II and CD86 expression in these dendritic cells, we could observe that whether caused by Plasmodium infection or by inoculation of TLR ligands, all the dendritic cells in these mice had acquired an activated phenotype. And one of the things that we learned through many years of studies in my own group and many other groups is that when dendritic cells undergo activation, they change their antigen processing and presentation activities from a phenotype in the resting state that is very good at capturing antigens to a phenotype where the dendritic cells are very poor at capturing antigen and very poor at presenting antigen, even the antigen that they have captured. And this just summarizes a number of changes that happens in the dendritic cells during the activation process, but the most relevant can be summarized like this. An activated dendritic cell refuses to present antigens encountered post-activation. Normally, this is not going to be a problem when you have a mild infection, because only a small number of dendritic cells will respond to that. You are always going to leave behind a sufficient number of dendritic cells ready to respond to the next infection. But if all your dendritic cells become activated, as is the case during SARS, you are going to run into the problem that you have run out of dendritic cells capable of responding to a challenge encountered soon after the systemic activation. This is good things to know, because this informs us of what was missing in order to cause a response in mice that had undergone systemic activation. We thought that it was dendritic cells presenting antigen. And indeed, if we took dendritic cells from a normal mouse that had not been, from a mouse that had received, where all the dendritic cells had been activated, and we coated these dendritic cells with peptides. So we loaded artificially these dendritic cells with the antigenic peptide that they couldn't present on their own. And we injected these cells into mice where the dendritic cells of this mouse had been systemically activated and therefore could not themselves present antigen. This restored the ability of the adaptive immune system of these mice to mount immune responses. This means that the bottleneck to the mounting of an adaptive immune response in these mice that suffered systemic activation, SARS, 12 hours ago, 9 hours ago, the blockade to their immune system is the ability of dendritic cells to present antigen. If you put dendritic cells presenting antigen in these mice, you overcome that problem. Now, dendritic cells are renewed continually in the body, and in measurements performed over 2 decades ago in the Shorman laboratory, it was determined that, for example, the dendritic cells in the spleen within 1 week, less than that, all the dendritic cells are renewed, and they are renewed from bone marrow precursors that are continually seeding all tissues in our bodies. So, if the problem of systemic activation is that all dendritic cells become activated, it should just be a matter of days before all the dendritic cells undergo death, because we know that that is what happens to dendritic cells even after their activation, and should be replaced by new dendritic cells that we hope would be able to respond normally to the next challenge. So if you survive this dangerous period of systemic activation of the dendritic cells following SIRS, do you return to homeostasis? Well, the answer is no. We know that there is patients who survive systemic inflammatory response syndrome become refractory to mount immune responses for an inordinate amount of time afterwards. We have been interested in this problem as well, using several models in mice that reflected situations in humans where we could infect mice and ask how good was their response subsequently and identify issues with their immune system in cases where they couldn't respond. And we have published a number of papers on this. One of the advantages of this series of lectures is that because everything is available on the web and you can stop the video at any time, you can check the papers and check the details of the study there. I'm going to summarize just the main conclusions. In this one, we described that following bacterial pneumonia in mice, a situation that causes a systemic severe inflammation in the lungs, there is a period where both dendritic cells and macrophages become incapable of responding to subsequent challenges. We call this dendritic cell and macrophage paralysis. A subsequent study by the same author of the first study, Antoine Roquilly, in collaboration with Jeremy Postman and Karina Senune in Nantes, helped us understand in more detail what are the mechanisms that cause the impairment or the paralysis of macrophages following the first infection. This was the establishment of environment in the lung triggered by engagement of the SIRP-alpha receptor on macrophages that caused resident dendritic cells in the lung to undergo epigenetic modifications that made them less capable of phagocytosing bacteria for a long period afterwards. Some of you may realize the similarities of what I'm describing here with the process of innate immune training, a process that is very prominent, of which there has been many publications in recent years. What is surprising, though, is that what the training model proposes, and it has been demonstrated, is that following an infection, innate cells become capable of responding better to a subsequent infection. This is the case in certain conditions with some stimuli. We also know that there are other conditions and other stimuli that cause an anti-training, what we call paralysis. This is the mechanisms that prevent macrophages and dendritic cells to respond to subsequent infections. I've already told you very briefly some of the mechanisms that are responsible for this in macrophages. What about dendritic cells? Can we demonstrate that there is paralysis of dendritic cells long-term as well? For this, I need to introduce the life cycle of dendritic cells, which is important to take into account whenever you compare it to the life cycle of macrophages, which is very different. Because dendritic cells, as I said before, are continually renewed from bone marrow precursors. This is a clear distinction from the life cycle of macrophages, which are self-renewed in the tissues. In some tissues from precursors that seeded the tissues early in life or even during embryonic development. In other tissues are macrophages that develop from monocytes recruited to the tissue, where those monocytes become macrophages, and that then self-renew and stay there, maintaining the macrophage population. Dendritic cells do not work like that. Dendritic cells are continually renewed from bone marrow precursors that occupy all the tissues in our bodies and where they undergo final differentiation. And what is important to know is that those precursors reach the organs where they will become dendritic cells with their full functional capacity not fully developed. Moreover, it has been shown in papers from the laboratory of Shorman and Wicks and our own that local signals in the tissues promote the acquisition of functions in those developing dendritic cells. In other words, the functions of dendritic cells are not hardwired. They are induced by cues received in the tissues. And this makes a lot of sense when you consider that dendritic cells have to seed and develop in the dendritic cell precursors need to seed and develop into dendritic cells in the whole body in areas that have very different conditions in terms of exposure to pathogens and commensal flora. Imagine dendritic cell precursors seeding the gut or seeding the spleen. The spleen is essentially an sterile organ. There is very little exposure to pathogens or to commensal flora or to their products there. Those dendritic cells are going to be exposed to steady-state signals in the spleen. Likewise, dendritic cell precursors in the gut are going to be exposed to the tissue signals that will cause the differentiation of these dendritic cells acquiring the functions that are appropriate for that tissue. And there is no reason to believe that the signals are going to be identical and that therefore the activity, the capacity of the dendritic cells to work in those tissues is going to be identical. We can say that in both tissues, the dendritic cells are going to develop, okay, they are going to work properly in those tissues, but not necessarily identical. And this is what we have described in a recent reviews we published last year in Annual Reviews in Immunology as spatiotemporal adaptations that affect both dendritic cells and macrophages. Here, I'm not using only the term spatial to refer to different anatomical locations, but also temporal. If dendritic cell precursors can develop into dendritic cells with different capacities according to the tissue, to the different environment or different tissues, it also makes sense that if the conditions of that tissue changes due to a prior infection, there will be a temporal change in the conditions of that tissue, and therefore the dendritic cell will undergo differentiation in a different way. This is what we described in our studies of dendritic cell paralysis in the lung. Following a severe infection, there was a change in the conditions of the tissue, in particular with recruitment of regulatory T cells and levels of TGF-beta that caused new dendritic cells being recruited to the lungs post-infection, caused their development into dendritic cells with a paralyzed phenotype. So you can put together those results with a model like this, where at the top represents what happens in a tissue like the lung in normal condition. But if you have a severe infection, you will undergo the systemic activation I told you, but there will be post-infection paralysis signals left over in the tissue that will cause new dendritic cells to develop into a paralyzed phenotype that will cause immunosuppression, incapacity to respond to a subsequent challenge. It's going to take several days, weeks, independent on the case, for those paralysis signals to be gradually replaced by the steady-state signals that will enable dendritic cell development to return to normal. And this is one of the reasons why we believe the severe systemic inflammatory response syndrome increases the susceptibility to secondary infections. Immunosuppression increases the susceptibility to hospital-acquired infections. from which it follows that if you prevent immunosuppression, you should increase the resistance to hospital-acquired infections. It's for this reason that we perform these studies, to try to find mechanisms that you may be able to exploit to reverse the paralysis that occurs post-systemic inflammatory response syndrome. And this is an example where Antoine showed that blocking the SIRP-alpha to prevent the establishment of an inflammatory, of a pro-paralysis environment may be a mechanism to restore immunocompetence in patients who undergo systemic inflammatory response syndrome. These studies are a challenge to perform in the lung because of the scarcity of dendritic cells. And that's why we have also gone back to the analysis of dendritic cells in the lung, which have a major advantage. In the spleen, sorry. Virtually all the splenic dendritic cells in a normal laboratory mouse are in a resting state. And virtually all can be activated synchronously by inoculation of a stimulus, like a malaria infection or a TLR ligand, as I showed you before. And this means that all those dendritic cells can also be synchronously replaced by new dendritic cells that will be exposed to the consequence of the systemic inflammatory response syndrome, which, as I showed you before, can be triggered by the injection, in this case, CPG or LPS. And we can then ask the question, do we induce paralysis in the spleen following this treatment? And the answer is yes. It takes us several weeks to recover the full immunocompetence of dendritic cells in the spleen following a single injection of CPG or LPS. And this allows us now to study what is wrong with those dendritic cells and ask, first of all, well, could it be that they continue to be activated even after they are renewed by new dendritic cell precursors? So is there a continuous activation of dendritic cells post-SIRS? The answer is no. The appearance of those dendritic cells that replace the ones that detected the presence of the stimulus is almost indistinguishable from the dendritic cells that existed before the initiation of the treatment. So there are subtle changes that happen in these dendritic cells that are not caused by that are not a systemic maintenance of activation. Just in case that you think that this is artificial of the effect of CPG, we have also examined what happens post-malaria infection. And this is important because we know that induction of immunity in malaria endemic areas is hard, not only against the malaria vaccine, or experimental malaria vaccines, but also against other types of vaccines against other pathogens. And we know the frustration of malaria immunologists, where they develop a vaccine that seems to induce very good responses in areas where malaria is not endemic, but those same vaccines don't seem to be as effective in areas where malaria is endemic. We are hypothesizing that one of the reasons for that is that the frequent exposure of the immune system of these individuals to malaria infection makes the immune system overall less responsive. We can reproduce this in vivo in mice by treating the mice with the malaria parasite I showed you before. This is a treatment that normally kills the mice because we are using a parasite that causes the death of the mice. But you can cure them the way that you would cure a human by using an agent against malaria, in this case, chloroquine. And in this situation, we can observe first the induction of SIRS, as I showed you before, by the malaria infection, but then a return to lack of serum cytokines, so a return to resting conditions, which, however, is, even though is reflective in the production of new dendritic cells that do not appear activated as the ones that were present at the time of the peak of malaria infection, retain their incapacity to present antigen. They are paralyzed. So it doesn't matter whether the stimulus is a TLR ligand, whether the stimulus is an infection with a real pathogen. If you induce systemic inflammation or an inflammation that is high enough to cause a change in the environment where dendritic cells develop, they may develop into cells unable to produce good immune responses. What is exactly what happens? The ability to study dendritic cells from mice in resting conditions and mice that have undergone SIRS enables us to study what is wrong with the dendritic cells. And for example, we can inject fluorescent beads coated with an antigen and purify and ask first, what is the efficiency of capture of these beads? And what we observe is that dendritic cells have lost the capacity, partially, the capacity to phagocytose these beads. But this is all. You can also now purify dendritic cells that have captured exactly a discrete amount of beads. And you can then compare the ability of those dendritic cells to present the exact same amount of antigen that is associated with the one bead that they have captured. And you do that comparison, even when you compare dendritic cells from resting or paralyzed dendritic cells from those mice over there, even if those dendritic cells have captured the same amount of antigen, the later ones, the ones that have been activated, that are paralyzed, are poor at presenting the antigen. You can do the experiment slightly different by first purifying the dendritic cells and then exposing them to the antigen ex vivo. And then you can ask, how well can you present a discrete amount of antigen in those conditions? And surprisingly, what we observe is that neither phagocytosis nor the ability to present that form of antigen is nearly as impeded as it is in vivo, which means that what is causing the paralysis is as much a change in the dendritic cells as restrictions imposed by the environment where the dendritic cells are trying to perform their activity. So paralysis is mostly caused by the effect of the environment in real time on the dendritic cells that come after a systemic inflammation. We are currently undertaking additional studies to compare, for example, the transcriptome of these dendritic cells, trying to understand in more detail what is wrong, what are the activities that are failing in these dendritic cells beyond antigen capture, to try to find strategies to overcome it and improve the immunocompetence of patients that are suffering immunoparalysis due to systemic inflammation. This is the end of this part of my talk. And these are, I think that I've mentioned most of the people who did the most important experiments. I will move now to a different subject that puts me closer to the type of research that we do in the Bio21 Institute, more related to biochemical and cell biological processes. I'm going to summarize the results of a paper that we published last year that describes a novel form of cooperation between B cells and dendritic cells. And that started as a study to characterize what changes in dendritic cells during activation. We described a number of processes that include the surface expression of antigen-presenting molecules, the expression of genes associated with antigen presentation, and the ubiquitination of MHC class II molecules. This is going to be the main protagonist of the main biochemical activity that I will focus on in the next series of slides. Ubiquitination enables dendritic cells to continually renew very fast the information that they provide in form of MHC class II peptide complexes, because by ubiquitinating MHC class II molecules, dendritic cells can quickly deliver to endosomal compartments for degradation the MHC class II peptide complexes they normally produce in resting conditions. When dendritic cells undergo activation because they have encountered antigen, they stop the ubiquitination of MHC class II molecules, and that promotes the accumulation of molecules that were synthesized, that were produced soon after detection of the pathogen, to provide a memory of the pathogen detected in the form of long-lived molecules on the surface. This stage of ubiquitination is thought to be a critical component of the changes that dendritic cells undergo during activation, and is performed by a highly specialized ubiquitin ligase, an E3 ligase, one of the hundreds of ligases that we express, that has the peculiarity that is anchored to membranes and has a specificity for MHC class II molecules and also for the co-receptor CD86 via transmembrane interactions. This ubiquitin ligase adds ubiquitin to a single lysine residue in MHC class II molecules, so that if you eliminate either the ubiquitin ligase or the residue in MHC class II responsible for that ubiquitination, you cannot detect the formation of ubiquitinated molecules as detected here by Western blot of immunoprecipitated MHC class II. If you eliminate any of those two components, resting dendritic cells that normally express low level of MHC class II molecules because they are ubiquitinated and degrading surface MHC class II, they express high levels of MHC class II because the ubiquitin ligase is no longer able to promote the elimination of surface molecules. And that has been described in a number of studies mentioned there in the slide. We have asked in a number of studies what are the consequences of this process on the function of dendritic cells, and our laboratory, and my laboratory in collaboration with the laboratory of Justin Minton here in Melbourne, and also the laboratory of Paul Roche, have published a number of studies that describe these consequences. For the rest of my talk, I'm going to talk about an unexpected consequence that we characterized after realizing something that happened in mice deficient in this ubiquitin ligase in March 1. These mice have fewer dendritic cells. And in addition to containing fewer dendritic cells, we observed that they also contain a large number of cells that look like dendritic cells because they express a number of molecules found in dendritic cells, but they didn't fully look like dendritic cells. When we analyzed in more detail the expression of proteins normally found in dendritic cells, we observed that they were expressed at intermediate levels. They did not express molecules found in other cells, like macrophages and T cells or NK cells, but they express molecules normally found in B cells, and indeed at levels that are normally found only on B cells. This raised the question, are these dendritic cells, B cells, or something in between? To ask this, we perform a transcriptomic comparison of dendritic cells and B cells from wild type and mutant mice. They look essentially the same. And then ask, what do these peculiar cells look like? Are they more like dendritic cells, more like B cells, something in between? They look overwhelmingly like B cells. In particular, like one of the two major types of B cells that exist in the spleen. You have follicular B cells, which are the ones that you normally find that you normally study, are those responsible for the induction for the production of antibodies. And then marginal zone B cells, which are more innate type of lymphocyte that secretes a polyfunctional antibody early in life. It protects you before you build up your B cell memory after birth, and that we call the marginal zone B cells. These cells, as the name indicates, are found in the marginal zone in the spleen, and our analysis identified our special cells as marginal zone B cells. These cells did not express at gene level the genes required that encode for the proteins we observe. This is gene expression for the proteins that we observe on the surface of the cells, which beg the question, how it comes that these B cells acquire these in-membrane proteins? There is a mechanism that can account for this, and that I have already mentioned at the beginning of my talk, trogocytosis. Trogocytosis is the process where one living cell obtains a fragment from another living cell. And one of the potential consequences of that process is that the cell that trogocytoses places the proteins from the membrane of the trogocytosed cells on its own membrane, a process that has received a number of names, including the more familiar name for immunologists is cross-dressing, a term that was coined by John Udell to describe a process where dendritic cells can cross-present MHC peptide complexes generated by other cells, in this case obtained by trogocytosis. And we determined that B cells incubated with dendritic cells can trogocytose as measured by their acquisition of fluorescent dyes placed on the dendritic cell surface, and that this trogocytic activity is enhanced drastically when dendritic cells do not express MARCH1 or cannot ubiquitinate their MHC class II molecules. This does not only enable the B cells to acquire lipid bilayer, but also a number of membrane proteins from the dendritic cell embedded in that lipid bilayer, providing a potential mechanism for how we end up with marginal zone B cells expressing on their surface dendritic cell markers. We hypothesized that via trogocytosis, marginal zone B cells obtain these proteins, and in the process, it caused the elimination of dendritic cells via trogoptosis when this process was exacerbated due to the upregulation, to the elimination of MARCH1 ubiquitination. So why would the absence of MARCH1 make the B cells more appetizing for marginal zone B cells trogocytosis? We hypothesized that would be the overexpression of some protein on the surface that induced the trogocytic event. This is something that we have been interested in for a number of years. What are the proteins that are regulated by MARCH1? To answer that question, we developed a pipeline for proteomic comparison of any two surface proteomes. We described this originally in a study of Elodie Segura 1 decade ago, that we applied to the comparison of the plasma membrane of dendritic cells directly purified from the spleens of wild type of MARCH1 deficient mice. And when you compare the surface proteome of these dendritic cells, you find thousands of proteins, most of which are expressed at similar levels, and then some proteins that have upregulated only MARCH1 dendritic cells as potential targets of MARCH1 ubiquitination. Reassuringly, this included the proteins that we knew are regulated by MARCH1, MHC class II and CD86, but also some new proteins. And the most prominent hit was complement C3. This was a very surprising result, which we had to verify by using a number of antibodies specific against complement that we used to validate the result, showing that in the surface of dendritic cells is decorated with complement C3, detected with different antibodies. We could also detect it by Western blot by immunoprecipitating MHC class 2 and then detecting C3, which was observed as a band of a molecular weight that corresponded to a species of a covalent of MHC class 2 covalently bound to a fragment of C3. Why a fragment of C3? This is where I could introduce you to the world of complement, shown in a simplified way in this cartoon. But I'm not going to show you the body of work that we published describing how complement ends on the surface of dendritic cells. I will just give you the take-home message. We found that in the steady state, complement C3 is activated in a way that can bind to the MHC class 2 alpha chain, in particular to the carbohydrate linked to the MHC class 2 alpha chain. This chain is a glycoprotein. This recruits a number of factors that leads to the conversion of the complement that you normally find in serum into a form that we call C3DG. This is a normal process that occurs in our bodies normally to inactivate complement to prevent the deleterious downstream effects that you can get when complement remains active on the surface of healthy cells. And what we observe is that these complexes, just like MHC class 2 molecules not complex to complement, undergo ubiquitination and are eliminated from the surface of the cells. This provided an explanation for the accumulation of complement on the surface of dendritic cells deficient in MARCH1 or that have a mutation in the lysine in MHC class 2. If you cannot eliminate the complex, you will accumulate C3 on the surface. So what is happening in the mice deficient in MARCH1 is that the increased expression of MHC class 2 molecules with complement on the surface is making the B cells more prone to capture, to interact with the dendritic cells and to trogocytose the B cells. But this requires another player. It requires a receptor on the surface of the B cell that will recognize this complex. We prove, I will skip this, that this happens in vitro, and we proved that this receptor is the complement receptor 2, a receptor that happens to be highly expressed in B cells. And we determined this because in vitro, as I said before, you can increase drastically the amount of trogocytosis that B cells can do on dendritic cells when the dendritic cells express high levels of MHC class 2 coated with complement on the surface, but this goes virtually to zero when the B cells are not able to recognize that complex on the surface of the dendritic cells. We proved that this happens in vivo because we analyze a series of mutant mice where essentially we have two different types of situations. Mutations that enable complement to be highly expressed on dendritic cells is recognized by complement and can be recognized by the complement receptor in B cells, caused a loss of dendritic cells and an increase in the number of trogocytic B cells. Conditions that eliminate either MHC class 2 or C3 or the receptor in B cells that recognizes this complex will prevent trogocytosis in vivo and will prevent the elimination of dendritic cells in vivo. So this is a process that is happening constitutively in the mice, we believe that also in humans, and that is exacerbated by the absence of elimination of MHC class 2 complexes from the surface of dendritic cells. What can be the benefit of this interaction? What can be the function of this interaction? Well, to answer this question, this relates to the function of the marginal zone B cells themselves. And the short answer is we do not really know yet, but we can speculate on two ideas based on what has been described marginal zone B cells can do. First, as shown by Jason's sister, marginal zone B cells can capture antigen and can transported to follicular B cells in the follicles and give the antigen to follicular B cells so that they can do their own job of inducing antibodies against this antigen. We propose that by capturing the membrane of dendritic cells via trogocytosis, marginal zone B cells expand their ability to provide antigen to the follicular B cells, because one of the things that dendritic cells do very good is to capture antigens with a number of receptors. Marginal zone B cells may obtain the antigen from the dendritic cells, or they may use receptors, trogocytosed, cross-dressing from the dendritic cells to obtain the antigens themselves and then give it to B cells. There is another potential, and is that even though marginal zone B cells are thought to work mostly independently of T cells, there are situations where they can also produce, be engaged in immune responses that require interaction with T cells in the similar way that follicular B cells do. This is what are called the extrafollicular T cell B cell immune responses. But what is not very clear is what is the capacity of marginal zone B cells to capture and present antigen. And we wonder if by obtaining MHC class II molecules from dendritic cells, marginal zone B cells may acquire capacities that they wouldn't have without trogocytosis. We tested this by injecting mice with an antibody that delivers an antigen to a surface receptor expressed on dendritic cells. And following this targeting, the MHC class II molecules of dendritic cells generate MHC class II complexes, which you can detect with an antibody that is specific for that MHC peptide combination. When we treat normal mice with this agent, we can see very clearly the complexes on the surface of dendritic cells and a little bit in other cells, unsurprisingly, because the receptor that we are targeting is only on this population of dendritic cells, on the CDC1s. If the mice have B cells engaged in high levels of trogocytosis, the dendritic cells produce many more complexes. This is because these dendritic cells express higher levels of MHC class II. But more importantly, the marginal zone B cells also express very high levels of the complexes. And we know that these complexes are obtained by the marginal zone B cells via trogocytosis, because if you maintain the ability of the dendritic cells to present that complexes, but eliminate the ability of the marginal zone B cells to trogocytose dendritic cells, the number of complexes on the marginal zone B cells decreases drastically. So this is a direct demonstration that in vivo, the MHC peptide complexes of the dendritic produced by dendritic cells can be transferred to marginal zone B cells. And at least in vitro, these complexes can be used by marginal zone B cells to activate T cells. So I think that I have demonstrated that trogocytosis enables marginal zone B cells to acquire DC functions. I don't think that we know fully yet what are the full range of functions those may be. And I will leave the conclusions there for you to read if you are viewing this, as you are viewing this on YouTube. I will just finish with acknowledging the main author of the study that described these results, mine are Justine Minton's PhD student, Patrick Schreck. Thank you for your attention. Thank you, Laura, and the rest of the organizers of IMMUNOTALKs for this opportunity. And thank you to this audience for your attention. Bye-bye.
Oh, I'm just on mute. Thank you so much, Jose. We really appreciate you participating in our series of Global IMMUNOTALKs. I'm sure our participants watching online have so many questions for you. And so I'm just going to share my screen and let everybody know how we can do that. So just getting that up there. Okay, everyone. So to ask Jose all your questions, which Jose has agreed to kindly answer for quite the period after giving this talk, go on to Twitter, search for the account Global Immunotalks as usual, find the tweet that says ask questions for Dr. Jose Villadangos here, and then reply to that tweet with the hashtag for Global Immuno using, Jose will be responding using the @ImmunoSpeaker account. And so hopefully there'll be lots of dialogue ongoing after this wonderful webinar. And just to remind you, next week, it'll be a sort of a double whammy in the Melbourne edition. We will have Professor William Heath or Bill Heath joining us next week. And so that just leaves me to thank Jose once again for such a fantastic talk. And we'll see you next time. Thanks so much.